Assalamu alaikum. In this lecture, we will learn about the MCQs regarding the carbohydrates, especially carbohydrates, but some biomolecules related MCQs can also be present here. So let's start. A student was asked to estimate the concentration of glucose in a solution using the Benedict's test. The student was provided with a 1 mole per decimeter cube glucose solution and was told to make a 0.6 mole per decimeter cube solution by proportional gradation. So, which row shows the correct volume of both 1 mole per decimeter cube glucose solution and distilled water to make the 0.6 mole per decimeter cube solution? We have 6 test tubes, and we will take from one to other, and then in this way so on we will follow the procedure, right? And then we will find out that to make the 1 mole per decimeter cube glucose solution, the 12 centimeter cube of volume of 1 mole per decimeter cube solution is required, and the 8 centimeter cube water is required, right? So if we will use these quantities, then we will be able to get the 0.6 mole per decimeter cube solution. A student carried out the test on the same volume of the 4 different solutions to investigate the presence of the protein, starch and the reducing sugars in the each. The results are shown in the table. Solution 1 gives blue color with the Benedict solution, and purple with the biunit reagent, and yellow with the iodine solution. It means that the sugars are not present in solution 1. Proteins are present and starch is absent. So check solution number 2 give the Benedict solution orange color. Somehow the sugars can be present. Biunit reagent gives pale purple. A few amount of proteins can be present. It gives a blue black, means the starch is present. And solution number 3 gives orange color with Benedict. A few amount of sugars can be present. Biunit reagent gives purple color, means the proteins are present. Iodine solution gives yellow, so no starch is present. It means that the reducing sugars are present. No protein is present here, and iodine solution yellow means the no starch is present here. So what conclusion can be drawn from these results? From these results we can expect that solution number 4 has high concentration of reducing sugars and no starch is present in here. So option D is the right answer. A sample of the milk is tested with the Benedict solution, and after boiling a yellow color is observed. It means that a few amount of reducing sugars can be present. So we can say option D here is the right answer. The diagram shows the results of the test from the 4 different solutions containing the biological molecules, which shows that the solution that contains only starch and protein. Which shows the solution that contains only starch and proteins. If biunit test gives the purple color, that is a positive biunit test, means that the proteins are present. And the iodine test gives blue black color, means the starch are present. So we have to find out that option which concludes both starch and protein. So option D here is the right answer. Step 1 to 4 are used to test for a non-reducing sugar. Put 5 centimeter cube of solution into a test tube, add few drops of acid and then neutralize with alkali, and then add the Benedict solution. When is the solution boiled? Solution is boiled between 2 and 3 steps. That is after the addition of the acid, solution is boiled, and then after the addition of Benedict solution it also requires boiling. So option B here is the right answer.